Hi guys, I'm going to talk about meiosis, I'm going to talk about mitosis, I'm going to talk about cytoskeletal elements, all in this lecture. So what is this? This is a karyotype. What genetic disease does this represent? Push pause. Push pause and answer the question. Today, I'd like you to understand the forces that drive the mitotic spindle and cytokinesis. So I'm going to talk about the cytoskeleton because I'd like you to understand the forces that drive mitosis, the mitotic spindle, how microtubules are involved, how actin filaments are involved in cytokinesis. I'd like you to understand how meiosis difference. I'd like you to also understand how meiosis is different from mitosis. Meiosis is different from mitosis, and I'd like you to understand that. Finally, I'd like you to appreciate the benefits of sex. We've talked about the cell division cycle. That is the ordered progression through DNA replication and then separation of chromosomes. Mitosis is M phase. That is just the green bit at the top, mitosis. If you've studied biology in high school, you've gone through mitosis, and there's a really good diagram in your book that explains all these different phases. I think that I probably don't need to go through them in gruesome detail. Prophase, prometaphase, metaphase to anaphase, telophase, cytokinesis, interphase. The cell cycle, G1S to G2, all that stuff is interphase. The metaphase to anaphase transition here is when the chromosomes are lined up and separate. I've talked about how separase cleaves cohesins so that the chromosomes can be pulled apart. That's governed by the cyclin B-CDK1 complex, the M cyclins. Now I'm going to talk about how the chromosomes are pulled apart. What are the roles of microtubules? So microtubules are part of the mitotic spindle. Microfilaments, actin filaments, are part of the contraction that separates the two cells during cytokinesis. And of course, there are motor proteins that are involved in both of those events. Each cytoskeletal element has its own motor. The mitotic spindle is this whole thing that is the microtubules, the microtubule organizing centers that have been duplicated, the chromosomes that are attached to them. That's the mitotic spindle. The cytokinesis part is contraction when the chromosomes have been pulled apart sufficiently so that two cells can form from one. Okay, cytokinesis, let's talk about that first. Cytokinesis is the final separation. It involves actin and myosin. Actin is the cytoskeletal element. Myosin is the motor that works on actin. A contractile ring forms of actin filaments. Here's a scanning electron micrograph. The contractile ring forms here. That's what this red bit is. Here's an electron micrograph showing that the actin filaments are concentrated right at this contractile ring. Cytokinesis involves actin filaments. Let's talk about actin filaments, okay? These are flexible filaments. They're throughout the cell, but they're concentrated in the cell cortex. So here's a polarized epithelial, three cells, and the actin filaments are concentrated around the cell cortex, the outside of the cell. They form bundles, meshwork, and gels, and they can be branched, and they're really highly dynamic. So they grow and shrink, and their growing and shrinking is governed by our old friend ATP, binding and hydrolysis. Okay. Importantly, they have polarity. There are some kinds of cytoskeletal elements called intermediate filaments that you should read about in chapter 17 that don't have polarity. Nuclear lamins are an example of intermediate filaments. Keratin is another example. These don't have polarity and they don't have motors. Actin filaments have polarity, so you can distinguish one end from the other, and you can have motors going towards one end or the other. So here's an actin filament. It's composed of subunits. In general, the subunits that are bound to ATP add to the growing end, and those that are bound to ADP dissociate more easily. Okay? ATP hydrolysis occurs right in the little subunits. Okay, so they sit there for a while, and then after a certain time, they hydrolyze their ATP and they have ADP bound. In this case, ATP is serving, like GTP and other things, as a molecular switching device. Changes conformation a little bit, makes it more easy for the subunits to dissociate. The motors are myosins. They're involved in cell motility, okay? Cells move throughout the body. You can put them on a dish and they'll move, okay? They extend little feet, right? Filopodia, that's little fingers. Lamellipodia, that's kind of sheet-like structures. These are lamellipodia. All of this is just filled with actin. Of course, you've heard of actin and myosin. That's the force required in muscle contraction. And finally, actin and myosin are involved in cytokinesis. So, that's microfilaments, actin filaments, and the myosin family. Let's talk about microtubules and the mitotic spindle. Here's a beautiful picture that was from Science 2007 showing the mitotic spindle with chromosomes attached. These red things are the kinetochores attached to kinetochore microtubules. These are the microtubule organizing centers, This is metaphase. So, during mitosis, the centrosome, that is the microtubule organizing center, duplicates and then separates. From the two separate. Each centrosome nucleates an aster of microtubules, a star of microtubules. And then the microtubules between the two start interacting with each other. Okay? Kind of keeps them apart. They also send microtubules out to the cell cortex, the edge of the cell, and that kind of keeps them localized in this arrangement of the spindle. So it duplicates and then pulls apart. And during this time, of course, the chromosomes are condensing and lining up. Let's talk about microtubules. Microtubules are way cool. They're hollow cylinders. Okay, they're not like actin filaments. They're stronger, but they're more easily broken. They're composed of mostly alpha and beta tubulin, as little dimers, and gamma tubulin is the nucleating center. They have polarity. They have motors. The minus ends, so they have a plus and minus end. The minus ends are attached to the centrosome, and the plus ends are always out. Okay, that's different from actin filaments. They can be any which way, every which way, and branched. Microtubules don't branch like actin filaments. They're highly dynamic. Their growth and shrinkage is highly regulated by a number of proteins. That's the same with actin filaments. They're highly dynamic during mitosis. So they're hollow cylinders. Alpha and beta tubulin form these head-to-head dimers. And this is a protofilament. And 13 filaments are arranged in a helical tube. So it's slightly offset. And it's hollow inside. There's water in there probably. Here's what it looks like by electron microscopy. So they really are cool things. At the microtubule organizing center, alpha, beta, and gamma tubulin are present. So gamma tubulin is this more stable structure. That forms the nucleation site for the growth of this hollow cylinder. Okay? It's called the gamma tubulin ring complex. It nucleates microtubule growth at the centrosome. Note that centrioles are these little cylinders that are kind of perpendicular to each other, centrioles. Nobody really knows what these do. They're often, but not always, localized with the centrosome, the microtubule organizing center. So they're not the same thing. Centrioles are not the same as the centrosome. Often, but not always, localized together. Microtubules grow and shrink dynamically. It's called dynamic instability. In this case, the molecular switch is GTP, not ATP like actin. Actin, ATP. Tubulin, GTP. So, similar to actin, GTP-bound subunits add, they're part of the growing end. So when there's a lot of GTP-bound alpha and beta tubulin around, they'll grow, microtubules will grow. Then, over time, they hydrolyze their GTP and it forms GDP. And then they start to shrink. In fact, they can shrink really fast. They can kind of peel away. And it's called catastrophe when they peel away. Catastrophe! Microtubule motors recognize the polarity. And in general, dyneins are the ones that move towards the minus end. So in nerve cells, microtubules are pointing with their plus ends out down the axon. And when vesicles are transported back from the axon tip on microtubules, dynein is the guy that moves them. Kinesins, most of the kinesins, move towards the plus end. Okay? So two classes of motors. They have lots of important functions, moving organelles around in cells. Microtubules also organize the Golgi complex during interphase. The Golgi complex flies apart into vesicles during mitosis. So I made this table of properties that are similar and different between microtubules and microfilaments, actin filaments. They have a nucleation complex that's related, so alpha-beta tubulin is the filament part. Alpha-beta tubulin is part of the hollow cylinder. The nucleation is gamma tubulin. For actin, the nucleation is an actin-related protein, ARP23, okay? Different, slightly different than the actin subunits that add. They have monomers, alpha-beta tubulin, monomers, actin. Their dynamics are regulated by nucleotide hydrolysis, but they have different nucleotides. Microtubules, GTP, actin filaments, ATP. Their localization is governed, in the case of microtubules, by the centrosome. Microtubules are plus end out. Actin filaments are localized in bundles. So if you think of it in terms of a building, a skyscraper, microtubules would be like the steel frame, and actin filaments would be like the wood frame around the steel skeleton. And they have different motors. Microtubules have kinesins and dyneins, and actin filaments have myosins. All right, let's talk about the mitotic spindle now. Now that we understand microtubules. Microtubules form from the centrosome and radiate from the centrosome out to the cell cortex. These are called the aster microtubules, right here. Microtubules from the two microtubule organizing centers interact and stabilize, and these are called interpolar microtubules. Okay, that's these purple ones. And finally, there is some microtubules that actually interact with the paired chromosomes with the kinetochore. Okay, that's this little red thing. Let's focus on that for a second. Let's look at the chromosome pair, the sister chromatids, with the kinetochore. Kinetochore is not always in the center, but it is a region, a highly complex assembly of proteins that forms during mitosis, during late prophase. It attaches to microtubules that are directly connected to the mitotic spindle. Okay? Kinetochore. During anaphase, there's two phases of anaphase. Anaphase phases. Anaphase phase A, the chromosomes are starting to be pulled apart, okay? That is right when they're just separating. When separates cleaves the cohesins, right? So what happens is there's motor proteins right at the kinetochore, and there's a depolymerization of microtubules there as well. So the motor proteins are pulling towards the aster, and they're depolymerizing the microtubules as well, really fast. When I was a kid, they used to have these big machines when there was a storm and the trees would fall down, they'd have to get rid of the trees. They'd have these big tree hoppers. They'd take whole trees in, shove them in, and the chips would fly everywhere and made a big noise and woke everybody up. Really noisy machine, but boy did that thing work fast. I think that the kinetochore acts a little bit like that. It's a microtubule hopper. They go like this, and the little subunits are flying out, and the microtubules are depolymerizing, and so the chromatids are moving towards the poles of the spindle. That's the first stage. The second stage is involves some of these other microtubules. So the microtubules that are attached to the cell cortex, they start pulling away, and the microtubules that are interacting with each other, the interpolar ones, they start pushing each other. So the net result is the two stars, the two asters, are being pushed and pulled away from each other, making two cells. Right? So then cytokinesis can come and pinch the membranes away from forming two cells. So that's mitosis. That's the dynamics of mitosis. In this way, one cell becomes two. Now, let's think back to sperm and egg, right? What happens when sperm meets egg? Cells divide and divide and divide and divide, and then what happens? You get a lot of babies. And all these babies are different from each other, right? Why is there such diversity? Why aren't we just clones? Look around the class. Look, everybody you see, we're all different. Even siblings are different from each other. The reason is because there are benefits to sex, okay? What are the benefits to sex? You mix two genomes. Instead of just cloning yourself, you mix two genomes so you can select, theoretically, for the best of both, right? The offspring are distinct from each other when there's this mixing, and they're distinct from their parents, okay? As opposed to budding like yeast, as opposed to being identical. This genetic diversity enhances survival in an unpredictable environment. Meiosis is the production of specialized cells for sex, okay? Meiosis. So you have parents that are diploid. That is, they have two copies of every chromosome. Meiosis produces cells that are haploid. That is, they have one copy of every chromosome. These then can fuse to form a diploid, creating a new organism. Totally new. So there's mixing of the two genomes. Sperm and eggs are called gametes. Okay, these are specialized cells for reproduction. And the germ cells are haploid. They have one copy of every chromosome. The fertilized egg then is called the zygote, and that's diploid. It has the DNA from both the sperm and the egg. Then the zygote divides and divides and divides, and that's all the mitosis and the cell division cycle and all the signaling that we were talking about before. Those are all somatic cells, as opposed to germ cells, okay? Germ cells and somatic cells. Meiosis happens in germ cells. They form early in development, pretty early actually, when the embryo is just starting to make different types of cells. So these are called germline cells, and they'll eventually produce a bunch of sperm or eggs. Sperm and eggs. These will form sperm and eggs, which then fuse to form a zygote. All right. Meiosis is different from mitosis. Chromosome separation occurs in two stages, meiotic division 1 and division 2. Cells go from having replicated their DNA, 4N, to diploid, 2N, to then N, haploid. So there's two divisions instead of one, and they don't replicate in between those, okay? Two divisions. Then what happens is you have homologous chromosomes pairing with each other, okay? And they can undergo recombination, and they can be sorted in different ways. So there's two ways of creating diversity. I'll talk about both of those. Recombination and sorting. So here are two of my friends holding up noodles, pool noodles, which are meant to represent chromosomes, okay? A normal somatic cell, before DNA replication, has 2N, okay? One chromosome from the mom, one from the dad. After DNA replication, the chromosomes have duplicated, so it has 2 chromosomes from the mom and 2 from the dad. And that's called 4N, tetraploid, because they have 4 copies of all the genes. During meiosis, these chromosomes form one big pair, a pair of pairs. Okay, so these are homologous chromosomes, 4 of them pairing together. In the first division, they're separated back, 2 cells are formed, and you have 2N in each cell. Okay? After meiotic division 2, you have 4 cells, and each now has just 1 copy of each chromosome. And these chromosomes can be mixed together. Could have, since you have 23 chromosomes, a handful could be from mom or from dad, right? So here's what it looks like in a diagram from your book. In mitotic cell division, you have DNA replication, so you go from 2N, diploid, to 4N, and the chromosomes pair up, and they are pulled apart. Each cell is identical to its mom. In meiosis, it's different. You have 4N after DNA replication, but then you got this chromosome pair, this bivalent, right? These can recombine with each other, and then they're pulled apart once, and then they're pulled apart again, so you have 4 cells, each with 1 copy of the DNA. Meiosis is different from mitosis. And then, of course, when you have sperm and egg, they fuse to form the zygote, which can go on to make another embryo. Why is this good? Because of diversity. We've been championing diversity in this class, and diversity is a good thing. you get genetic diversity from sorting different combinations of chromosomes. So you get a handful from mom, a handful from dad, right? During that first stage. And you get crossover during meiosis to give hybrid chromosomes, okay? So chromosomes that contain parts of mom and parts of dad. And then these are sorted in different combinations. So it shuffles the genes around, shuffles the DNA around, okay? So in mitosis, the chromosomes are paired. Here's the asters, right? These are all lined up. Each of the daughter cells will receive an identical chromosome. But in meiosis, that doesn't happen. You might have the maternal ones going to one side and the paternal ones going to the other side, okay? So it's fundamentally different at this first stage. These pairs are called a bivalent. So the chromosomes are paired, and the legs can go cross over to each other, right? And so you get exchange. And so here's a picture of an actual chromosome by electron microscopy. And you can see this DNA strand is crossed over to this one and vice versa. So here's what it looks like when you have red ones and blue ones. You can see they're mixing up their origins, shuffling the DNA. These crossovers are called chiasma, and chiasmata is the plural of chiasma. These are the crossover sites. These help hold the bivalents together, and when a force is generated by the microtubule, similar to as in mitosis, then these are broken. But they're only broken when the kinetochores are attached to microtubules. And then you have chromosomes with parts from mom and parts from dad. Okay? Diversity. Diversity is good. Then, during meiotic division 2, the kinetochores are attached to microtubules of opposite spindle poles, as in mitosis. Cohesins are degraded, as in mitosis, and the sister chromatids are pulled apart. But there's only one per cell instead of two. Okay? And again, you might have bits from crossover being shuffled around. So, crossover can shuffle the DNA on individual chromosomes. Individual chromosomes can also be sorted in different combinations. So let's imagine a simple organism here with 6 different parental chromosomes, okay? If you can see the colors, you've got red, yellow, brown, dark brown, blue, and green, okay? During meiosis, you only get one of each of these, but you might get one yellow one green one brown one, you might get one yellow one blue one brown one. You have all different possibilities for combinations for mother versus father, right? So you have many possible gametes. Just from this pair of 6 here, you can have 8 possibilities. The combinations are endless. Then, for each one of these, you can have, in addition, you can have this crossing over that I was telling you about before. So these can contain bits from mom and bits from dad. So it's interesting to consider that while it's certain that you have equal amounts of DNA from your mother and your father, because you have one gamete, sperm, and egg from each parent, right? You have equal DNA from mother and father. The same is not true for your grandparents. Because of this and this, you may have more of one grandparent versus another. So your parents, you're equal, but your grandparents, you're not. I think that's interesting to consider. That's why everybody looks so different. Things go wrong, cause cancer, cause Down syndrome. When chromosomes are not sorted properly, you can have the incorrect number of chromosomes. That happens when one cell doesn't get any of one particular chromosome and the other cell gets 2 Okay? In contrast, where you're supposed to get one of each. So that looks like this when we have our friends with the noodles. You have a non-disjunction event causing aneuploidy. Here's the normal case. This guy doesn't have this orange chromosome, and this one has 2 copies of it. You get an extra copy of that particular chromosome. Then, when you have sperm and egg fuse, if you got the extra copy, you got 2 copies of the orange one, where you're only supposed to have 1, compared to the green one where you have 1. Okay? This is not good. This is what causes Down syndrome. So the answer to the question I posed at the beginning is that this is a karyotype from trisomy 21, Down syndrome. Similarly, if you have a normal cell fusing with a cell that's missing a chromosome, then you'll have only 1 copy of the chromosome. And if you're missing a whole chromosome, that's not good. That leads to the end.